Welcome, guys. In this session, we'll be looking at regulation of gene expression in prokaryotes. We'll start with an overview, then go to the regulatory, look about regulatory sequences and molecules, then regulation of prokaryotic gene expression. Then we look at, under that, we look at Lac Operon and the tryptophan operon. Welcome all, my name is Wallace Munir, and I'm the redactor. Now, let's start with this brief overview. What is gene expression? This is the multi-step process that ultimately results in the production of functional gene product, either RNA or protein. The first step in gene expression is the use of DNA for the synthesis of RNA, which you know is known as transcription, and it is the primary site of regulation in both prokaryotes and eukaryotes. Now, in eukaryotes, however, gene expression also involves extensive post-transcriptional and post-translational processes, as well as action that influence access to particular regions of a DNA. Okay, that is in prokaryotes only. In eukaryotes, this is the primary site. Regulation of transcription is a primary site for gene expression. Now, each of these steps can be regulated to provide additional control over the kinds and the amounts of functional products that will be produced. Not all genes are tightly regulated. For example, genes described as constitutive. So constitutive genes are those genes which encode products required for basic cellular functions and are expressed at essentially a constant level, okay? They are also known as the housekeeping genes. So constitutive stroke housekeeping genes, they are not tightly regulated, okay? But we have other genes known as regulated genes. They are expressed only under certain conditions. They may be expressed in all cells or in a certain subset of cells. For example, hepatocytes, right? We know hepatocytes are the only cells in the body which can synthesize ketone bodies. So if you require ketone bodies, remember one enzyme which is required in the formation of ketone bodies. Remember one enzyme. So only the genes in the hepatocytes are turned on for the synthesis of those enzymes. But for the others, no. But for extrahepatic cells, so the genes which synthesize the enzyme which is required for the utilization of ketone bodies is turned off in the hepatocytes, but it is turned on in the other cells. Now, the ability to regulate gene expression, that is to determine if, how much, and when a particular gene product will be made, gives the cell control over structure and function. And this forms the basis of cellular differentiation, morphogenesis, and adaptability of any organism. Now, control of gene expression is best understood in prokaryotes, but we have many themes which are repeated in eukaryotes. So first understand the prokaryotes, then move on to the eukaryotes. This is what I was saying. Transcription, it is the main site of control. Sorry, did not intend that to happen. So for most genes, the main site of control is the transcription of DNA to RNA, okay? But in eukaryotes, gene expression is also controlled at post-transcriptional and post-translation processes. Let's look at regulatory. Sorry, my cursor has started to misbehave. Let's look at regulatory sequences and molecules. Now, regulation of transcription, which is the initial step in all gene expression, is controlled by regulatory sequences of DNA that are usually embedded in the non-coded regions of the genome. Now, the interaction between these DNA sequences and the regulatory molecules, one example being transcriptional factors, can induce or replace the transcriptional machinery, influencing the kinds and the amount of products that will be produced. Now, the regulatory sequences are known as cis-actin. Why? They influence expression of genes on the same chromosome as they are found on. okay? But the regulatory molecules, which are mainly proteins, are known as transactin because they can transist or they can diffuse through the cell from the site of synthesis to DNA binding sites. For example, a protein, which is a transcription factor, can regulate a gene on chromosome number 6, but itself was produced from a gene on chromosome number 11. So it is not regulating gene expression on the same chromosome that it was produced from. So it is diffusing, it is transisting through the cell to regulate gene expression of another gene on another chromosome. So it is transacting. Now, these regulatory molecules, mainly proteins, they will bind to the DNA through structural motifs such as a zinc finger, a leucine zipper, a helix turn helix in the protein itself. So that is, here you can see, here we have, okay, my cursor again has started misbehave. Here you have now the cis-actin elements, and you can see it will regulate gene expression on the same chromosome as it is found on, but transactin factors, they can be synthesized from a different region, okay, and will target genes on different regions or different chromosomes than those that synthesize them. Okay. So here you can see one of the structural motifs used by proteins to bind to the DNA. This is a zinc finger. So regulation of prokaryotic gene expression. In prokaryotes such as your E. coli, regulation of gene expression primarily occurs at the level of transcription. Please understand that. And it is mediated by binding of transactin proteins to cis-actin regulatory sequences on the single DNA molecule. Note this, regulating the first step in expression of gene is a very efficient approach because you will not waste energy making unneeded gene products. Okay, so why should the hepatocytes, take this as an example, why should the hepatocytes synthesize that enzyme which is required for ketone body utilization if they will not utilize the ketone bodies? So you don't need to waste energy on something which you won't use, okay? So transcriptional control in prokaryotes can also involve initiation or premature termination of transcription. So this premature termination of transcription, I will show you which is used in the tryptophan operon. The process known as attenuation. We will look at that later. Messenger RNA transcription from bacterial operons. In bacteria, structural genes that encode proteins involved in a particular metabolic pathway are often found sequentially grouped on a chromosome, along with their cis-actin elements that regulate the transcription of these genes. The transcription product is a single polycistronic messenger RNA. Okay, so these genes will be regulated because they have a cis-actin elements. These genes and those genes are the ones encoding proteins which will be involved in a particular metabolic pathway, and they are sequentially grouped on a chromosome. Those genes will be either turned on or off as a unit. And that entire package is what now I'm referring to as an operon. So these operon have to be operated by operators. Now, bacterial operons contain an operator, which is a segment of DNA that regulates the activity of the structural genes of the operon by reversibly binding to a protein known as a repressor. If the operator is not bound by a repressor, RNA polymerase will bind to the promoter, pass over the operator, and reach the protein coding genes that transcribes an messenger RNA. However, if a repressor is bound to the operator, the polymerase is blocked and does not produce messenger RNA. So as long as the repressor is bound to the operator, no messenger RNA and therefore no proteins will be made. However, when an inducer molecule, so inducer molecule usually bind to the repressor. And once bound to the repressor, they will cause a conformational change to the DNA binding domain of the repressor protein. Hence, the repressor protein cannot bind to the operator. And if once the repressor operator, the repressor protein is not bound to the operator, the RNA polymerase will initiate transcription. So let me, okay. So I'm trying to draw a DNA sequence here. So let me divide it into some parts. So this is the promoter region. This is the operator, and these are now the structural genes. Let's say for lactose, Lac Z, Lac Y, and the Lac A genes. So RNA polymerase usually bind to the promoter region with the help of the sigma factor. Then if the operator site is clear, is unbound by the repressor, it will proceed and carry out transcription. But if the repressor is here, is bound, it cannot, there will be no transcription, so no protein formation. But if we have inducer molecules, these inducers will bind to this repressor. Once bound to the repressor, they will cause a conformational change in the DNA binding domain of this repressor, causing it to be unbound from the operator. Once unbound from the operator, the RNA polymerase will now carry out its transcription. That's what that page is trying to say. Now, let's look at the Lac Operon, which contains the genes that code for 3 proteins which are involved in the catabolism of lactose. Now, one of these structural gene is Lac Z gene, which will code for beta-galactosidase, which hydrolyzes lactose to galactose and glucose. Lac Y gene, which will code for permease. This permease will facilitate the movement of lactose from the environment into the cell. Then the Lac A gene, which will code for the thiogalactoside transacetylase, which acetylates lactose, though we don't know the physiologic function of this acetylation. All these proteins are maximally produced when lactose is available to the cell and glucose is not. So please understand that first. Now, bacteria use glucose, if available, as a fuel in preference to any other sugar. Please also understand that they will choose glucose over any other sugar. Okay, so even if you give them maltose, arabinose, still choose glucose. Okay, so the regulatory portion of the operon is upstream of the 3 structural genes and consists of the promoter region, where the RNA polymerase binds, and 2 additional sites. One site is the operator, where the repressor protein will bind. We also have another site known as the catabolite activator protein site, the CAP site, or the cyclic AMP regulator proteins site, CRP site, where now what binds to the CRP, the CAP site, is the cyclic AMP CRP complex. Okay, so those are the regulatory proteins. Now, Lac ZYA genes are maximally expressed only when, please understand this statement, O site is empty, so the repressor is not bound to the O site, and when the CAP site or the CRP site is bound by a complex of cyclic AMP and CAP, the catabolite activator protein is also known as cyclic AMP regulatory protein. So that is when those genes will be maximally expressed. Now, a regulatory gene known as the Lac I gene will code for the repressor protein, which is a transacting factor, and binds to the O site with a very high affinity. Okay, note that Lac I gene has its own promoter, and it is not part of the Lac Operon. So Lac Operon has 3 structural genes, a CAP site, a promoter site, and an O site, or the operator site. Lac I is not part of the Lac Operon. In fact, LAC-I gene itself is not a regulated gene. It is a housekeeping stroke constitutive gene. So now let's look what happens. How is the Lac Operon regulated under these three conditions? Condition number one, glucose is available. Condition number two, only lactose is available. Condition number three, both glucose and lactose are present. So let's start with condition number one, when only glucose is available. In this case, the Lac Operon will be turned off. It is repressed. This repression will be mediated by the repressor protein from the LAC-I gene, and it will bind via a helix motif to the oocyte. Okay, so its DNA binding domain has a helix motif. Okay, to the operator site. To bind via that motif, using that motif to the operator site, which is downstream to the promoter. So binding of the repressor interferes with the binding of RNA polymerase to the promoter, and this will inhibit transcription of the structural genes. And this is one example of negative regulation. What about when only lactose is available? In this case, the Lac Operon is maximally expressed, stroke induced, stroke turned on. A small amount of lactose is usually converted to allolactose, which is an isomer of lactose. This compound will act as the inducer. Allolactose now will bind to the repressor. Once bound to the repressor, it will change its conformation so that it can no longer bind to the oocyte. Okay. Now, in absence of glucose, that is when only lactose is present, adenylyl cyclase is active, and it will leave ATP to form cyclic AMP. Then this cyclic AMP will bind to the catabolite activator protein, or the CRP. Now, then this complex, the cyclic AMP cap transacting complex, will bind to the capsite. And once bound to the capsite, the RNA polymerase will initiate transcription with a very high efficiency, okay, at the promoter site. This is an example of positive regulation. Then the transcript made will be a single polycistronic messenger RNA that will contain 3 sets of start and stop codons. Okay, and now translation of the messenger RNA will produce the 3 proteins that allow lactose to be used for energy production by the cell. This is what I was saying. LAC-I gene is constitutive. Its gene product, the repressor protein, is always made. But so you have constant production of the repressor protein, but its activity will be determined by presence or absence of allolactose. So it is inactive when allolactose is present, but active when allolactose is absent. So when allolactose is not present, it will bind to the oocyte, but when allolactose is present, it will, the allolactose will bind to the repressor, changing its conformation, hence it will no longer bind to the oocyte. What about when both the glucose and lactose are present? The Lac Operon will still be uninduced. Okay? Why? And transcription will be negligible even if lactose is present in high concentration. I know you're asking yourself, we have lactose, so it will be isomerized to allolactose, and this allolactose is acting as an inducer, which will cause conformational change to the repressor protein. So the oocyte will be unoccupied, which is very correct. But remember, glucose is present. Glucose presence means inhibition to the adenylyl cyclase. Okay, so no cyclic AMP will be formed. So, in fact, that process where glucose inhibits adenylyl cyclase is known as catabolite repression. Okay, so no cyclic AMP cap complex forms, and thus the capsite will remain empty. So RNA polymerase is unable to effectively initiate transcription, even though the repressor is not bound to the oocyte. So it can bind to the promoter site, but it will not carry out transcription since the capsite is empty. So this is what I'm saying. So in this case, we have glucose present, only the glucose present. The operon is switched off, turned off, because first of all, we will have no inducer. No inducer present, so the repressor protein will be bound to the operator. Also, glucose is present, so means that adenylyl cyclase will also be inhibited. So no formation of the CAP, CAP, no formation of the cyclic AMP CAP complex. So also the CAP site is unbound. What about when only lactose is present? When lactose is present, adenylyl cyclase will be very active. So formation of cyclic AMP will bind to the CAP protein. So that CAP cyclic AMP complex will then bind to the CAP site, okay? Then some of the lactose present will be isomerized to allolactose, which acts as an inducer. Once bound to the repressor protein, it will the repressor protein undergoes conformational change, can no longer bind to the O site, and once that is done, the RNA polymerase will initiate transcription. Okay. What about when both are present? If both are present, yes, some of the lactose will be isomerized to allolactose, and hence the operator site will be unoccupied. But since glucose is present, adenylyl cyclase activity will be inhibited, so there will be no formation of cyclic AMP CAP complex. So the CAP site will be empty. So the RNA polymerase cannot effectively initiate transcription. So there'll be very low levels of messenger RNA and therefore protein expression. So please, you can pause at this point, look at that. This is a very helpful. It summarizes the Lac Operon. Now this is the helix motif of the Lac repressor gene. This is the structure which the Lac repressor protein uses to bind to the oocytes. Now let's look at tryptophan operon, which consists of which contains 5 structural genes that code for enzymes required for the synthesis of the amino acid tryptophan. Now, please understand this about tryptophan operon. As with the Lac operon, tryptophan operon is also subject to the negative control. However, for the repressible tryptophan operon, negative control will include tryptophan itself binding to the repressor protein and facilitating the binding of the repressor to the operator. So tryptophan in this case is a co-repressor, not an inducer, as in the case of allolactose, which is an isomer of lactose. So in this case, because repression by tryptophan is not always complete, the tryptophan operon, unlike the Lac operon, is also regulated by a process known as attenuation. With attenuation, transcription is initiated, but is terminated well before completion. So you initiate transcription, then before it is completed, you tell it halt. Okay. So if tryptophan is plentiful, transcription initiation that escaped repression by the tryptophan, remember this is what tryptophan does. I don't know if I can draw. So let's say this is a case of, this is the DNA. Then here you also have a promoter site, an O site. Then the 5, 1, 2, 3, 4, 1, 2, 3. Yes, the TRP genes. They are the TRPE up to A. Okay. So if tryptophan is in plenty, okay, if tryptophan is plentiful, this is what occurs. So this is the repressor. Okay. So if tryptophan is present, it will bind to the repressor, and once bound to the repressor, this repressor will bind now to the O site. So no transcription of these structural genes. But if tryptophan is absent, the repressor will stop binding, will not bind to the operator site. Hence, there will be transcription of these genes, which code for the enzymes required for the biosynthesis of amino acid tryptophan. So if that did not occur, and the transcription initiation that which escaped repression by tryptophan is, if tryptophan is plentiful, transcription initiation that escaped repression by this tryptophan is usually attenuated or stopped by the formation of an attenuator. This attenuator is a hairpin structure in the messenger RNA, similar to that seen in the raw independent termination. Remember that in the raw independent termination, where you remember the termination of transcription, where we had two types, raw dependent, raw independent. And remember in the raw independent, we had formation of a hairpin structure. Okay, the same applies here. So we have a region of this operon, which is known as the tryptophan reader, TRPL. Okay, in fact, it occurs somewhere after I think it occurs somewhere after between the operator and the TRYP-E gene. So between the operator and the TRYP-E gene, we have a region known as the tryptophan reader, TRPL. So that region has sequences that are that can form close links. Okay, so it is not hard, but when we translate that messenger RNA, of this region of tryptophan reader, it will have a matching region. So let's say this is that region. So this is the messenger RNA transcribed from the TRPL, the tryptophan reader. So it will have regions which can cross-link. So it is usually divided into 4 parts. So region 1, region 2, region 3, region 4. Now, in absence of, so let's start in tryptophan plentiful. When tryptophan is plentiful, remember, in prokaryotes, both transcription and translation can occur at the same time. So if tryptophan is present, the first two codons here, the first two codons in region 1 are UGG. Remember, UGG we code for tryptophan only. Tryptophan and methionine are the only amino acids which are coded by one codon. For methionine is AUG and tryptophan is UGG. Okay, so if tryptophan is present, the messenger, the ribosome, the transcriptional machinery, the translation machinery will come and translate, okay? And they will do so. As they do so, there will be formation of stem loops, or this hairpin between region 1 and region 2, okay? And this, once this hairpin is formed, it will allow another hairpin to be formed between region 3 and region 4. But once the transcriptional machinery sees into brackets, in quotes, once it sees this hairpin between region 3 and region 4, it will terminate. It will terminate that. The transcription will be terminated. Okay, not the transcription, the translation will be terminated. Okay, so, but if tryptophan is not present, so the ribosomes, the transcriptional, the translational, sorry, I don't know why I'm saying transcriptional, the translational machinery will bind, two ribosomes will be bound to this region, UGG, the two UGG codons, because tryptophan is not present, and hence the amino acid transfer RNA for tryptophan is not present, there will not there be no formation of a stem loop between region 1 and region 2. Only one loop will be formed between region 2 and region 3, and that stem loop between region 2 and region 3 will now allow the messenger RNA, a full length messenger RNA will be transcribed. But if there is a stem loop between region 3 and region 4, there will be formation of a truncated messenger RNA. Now, because transcription and translation are temporarily linked in prokaryotes, attenuation also results in formation of a truncated non-functional peptide product that is usually rapidly degraded. Now, the truncated messenger RNA is rapidly degraded, but the full length messenger RNA is the only one which will be, we now go to the formation of the proteins needed for tryptophan biosynthesis. However, if tryptophan is scarce, the operon is usually expressed, and I have tried to explain how that will occur. Now, the 5' end of the messenger RNA will contain two adjacent codons for tryptophan, the UGGs. Now, the lack of tryptophan will cause ribosomes. Ribosomes will stall because remember, you cannot continue with the translation if you don't have that, the amino acid transfer RNA for that amino acid. So the ribosomes will stall at these codons, covering the regions of the messenger RNA required for the formation of the attenuation hairpin. The regions of the messenger RNA required for the formation of the attenuation hairpin are region 3 and region 4. So there'll be only formation of a stem loop between region 2 and region 3. Okay, so for when tryptophan is present, these ribosomes will proceed with the translation, but there will be formation of a stem loop or the hairpin between region 1 and region 2, then that will allow formation of another stem loop between region 3 and region 4. Now, when that translation machinery sees, or the transcriptional machinery sees the stem loop between region 3 and region 4, it will stop, it will attenuate, okay? And this will be done prematurely. So there will be formation of a truncated messenger RNA, which will be degraded. Now, but when we don't have tryptophan, there will be formation of a stem loop between region 2 and region 3, and this will prevent attenuation and will allow transcription to continue. Okay, so this is what this now is the now this is region 3 here and region 4. Once those regions have self-complementary sequences, okay, and will allow formation of a hairpin structure, which attenuates or prematurely terminates transcription. Okay, so the stem loop which is needed for the attenuation is that between region 3 and region 4, but region 2 and region 3 stem loop will allow formation of a full length messenger RNA. But for us to form a stem loop between region 3 and region 4, you must have formed another stem loop between region 1 and region 2. And this stem loop will only be formed when tryptophan is plentiful. But this will only be formed when there is no tryptophan. So the ribosomes will stop at the 5' end of the messenger RNA. So I think we are done, guys. So thank you for your attention, and subscribe for more biochemistry content. Thank you. So, okay, how was this? Okay, sorry.